Good evening. Good evening. And welcome to the Shoals Marine Laboratory's annual weekly seminar series, our Rock Talk series. I'm Sarah Morris, the John M. Kingsbury Executive Director of the Shoals Marine Lab, and I would like to thank everyone for joining us this evening for our 10th Rock Talk of our 60th season of the Shoals Marine Lab. For those of you who are joining us online, or for anyone who is new to our community, the Shoals Marine Lab is the largest and oldest organization as a marine laboratory dedicated to undergraduate development, both in terms of coursework and in terms of research. Our seminars are affectionately called Rock Talks because back in the early days of the lab, before we had buildings that had roofs, we would actually have our lectures out on the rocks as the students were getting ready to go into the intertidal. Now that we are all gathered together in Kiggins Commons, we still call them Rock Talks as a time for our entire community to come together and learn more about marine sciences. In addition to our guests joining us remotely tonight, we have 3 classes on the island. We have shark biology and conservation. Yeah! We have research in biology. Okay, shark people, help the other classes out, please. And we have underwater research. We also have 12 undergraduate interns, our Shoals undergraduate research group, who are conducting independent research here throughout the summer. We're excited to have everyone here, both in Kiggins Commons and online, joining us this evening. The format of tonight's seminar is a 45-minute talk followed by 15 minutes of questions and answers. For our friends online, we ask that you use the Q&A button at the bottom of your screen, and we will be able to read your questions out loud on the microphone so everyone in the room can hear us. I will be passing the microphone around for anybody that is here with us in Kiggins Commons. This evening's speaker is Brad Weiler. Brad is a Canadian postdoctoral researcher at the University of Miami's Cooperative Institute for Marine and Atmospheric Studies. Brad graduated with his BS in biology from Carleton University, focusing on benthic community phase shifts and disease ecology in Caribbean coral reefs in the Bahamas. He continued studying that system to assess coral reef health during the 2014 and 2017 global bleaching event. Brad received his master's in science in biology at Memorial University in Newfoundland, focusing on the prokaryotic communities found in deep sea soft corals. He earned his doctorate from the University of Miami's Rosenstiel School for Marine, Atmospheric, and Earth Science. He primarily investigates tropical coral holobionts, host and collective microbiome, using molecular biology omics tools such as metabarcoding and transcriptomics. His PhD dissertation was Understanding Marine Health and Disease, investigating healthy corals over natural diel cycles to assess their health at the molecular level and how disease impacts normal coral stress and disease. His goals are to better understand the cellular response mechanisms to disease and to help mitigate future outbreaks. This evening, Brad will be presenting, and I love the title as a flashback to Harry Potter. For those of you that don't know, I'm a Harry Potter fanatic. Brad will be presenting Microscopic Beasts and How to Find Them. Molecular surveys reveal hidden marine biodiversity from the microcosmos. Please join me in welcoming Brad Weiler.
Thank you very much for that lovely introduction. I know a lot of those words are full, so I hope I can clear the air on what those mean. So yeah, as you all know now, my name is Brad Weiler. I am a postdoc at, it's actually the Atlantic Oceanographic Meteorological Laboratory. It's in Virginia Key, it's Miami, part of UM. And today I'm going to go over kind of like a narrative of microbes, how they originated, how we started studying them, and kind of going through the tools as just like a nice story. And so I'm not going to be diving too deep into data, but please find me later and we can go over some burning questions that you might have. So to begin, I think it's important to clear some of this terminology that can be a bit of a mouthful. So the microbiome is a collection of the prokaryotic, so our bacteria and archaea, and our microeukaryotic communities, fungi, we have our protists, other algae,
and our viruses. These are the oldest occupants of Earth. They are literally everywhere, and they occupy all three domains of life. So this tree was developed by Laura Hugg's lab, and it kind of depicts a really nice just breadth of the biodiversity that bacteria actually encompass. And then just kind of going down the tree, we have our archaea. It goes into our Asgard archaea. And then just off to the right there, we have the green branch, and that is the Epidendronta. You are here. This is all the animals you know. Every animal, the gulls, they all fall under Epidendronta. So look at everything else on that tree. It's pretty stark. These microbes comprise over 90% of biological diversity on Earth. So it's pretty outstanding. This is a eukaryotic tree of life. So when I point at that little green branch, this is what it actually looks like. And so it is extremely diverse with wildly different survival mechanisms and lifestyles. Microbes are just unbelievable in how they're able to occupy very strange environments. So why study marine microbes? Oceans are about 70% of the Earth's surface. It's pretty substantial. And so microbes are shaping all the biogeochemical cycles: nitrogen, carbon, sulfur, and also consider our oxygen. People typically look to our rainforests, but over about 50%, I think, of the Earth's oxygen is actually produced by this little guy, Prochlorococcus. So right here is a global distribution of chlorophyll A concentrations, and you can see that they're typically spread towards the poles and then kind of hugging all the continents, all the edges. And so before we get into the crazy lifestyles and how to find them, it's important to understand how we got here, because molecular surveys are really impressive and we use these terms, transcriptomics and metabolomics and all that, but really it all started with convex glass. And so Van Leeuwenhoek, Antoine, he had taken two pieces of convex glass and developed the first microscope. Then described invisible life as animalcules, animal molecules, and found them pretty wildly confusing because they always assumed molecules and these looked like they were living. And so this was an absolute scientific revolution of its time. And so you might recognize this name, Louis Pasteur. He created the first liquid bacterial culture medium. This was nearly 200 years later. You might recognize the name from pasteurization. So Louis ended up pioneering this idea of taking microbes from our surrounding environment, bringing them in the lab and actually growing them. And that was, again, revolutionary. Fanny Hess was a microbiologist, very close with Robert Koch, one of his grad students' wives. She was an incredible microbiologist, and she was the first to suggest using seaweed derivatives to solidify that media at room temperature. Absolutely revolutionary. And so that's what we commonly know today as agar. And then Robert Koch, he had developed these set of criteria in which today we know as like how to identify an etiological agent. This is something that is pathogenic that causes a disease. And so there's a set of rules to follow essentially. But again, these are the absolute giants of our field. And so there is an issue, a bias in culturing. It's called the Great Plate Count Anomaly. So we have our environmental samples, some soil, some seawater. We throw it on the microscope, we see it's teeming with life, all these little critters running around, and we go to culture and we get splat. Only roughly 1, maybe 2% of known bacteria is culturable. Now, this doesn't mean that we can't culture it. It means we don't have the biological information to replicate the environments that they thrive in. And so this basically does not equal true diversity, right? We see all these microbes underneath the scope, and then we look at our agar plate, and there's maybe a couple of bacteria. And so we need cultivation-free tools, a way to explore these things without replicating their environment. And so how do we study what we can't grow? We use molecular methods, but they hadn't been invented yet. And so what we can do is we can, one, go through the culturing method, get to the end, take our little microbe, we can sequence it, get its genome, or we could do an uncultivated sequencing method. Just take that environmental sample. It's called metagenomics. I'll get into it. And you can assemble metagenome, assemble genomes. Bunch of fun terminology. Okay, so fast forward, it's about 100 years now, yeah, 1860. Frederick Sanger unveils his new pioneering technique, DNA sequencing, Sanger sequencing. And so up until this point, people were just able to basically take microbes, culture them, and look at them under the scope. Hope you do, right? So at this point, we have a way to start taking biological life and seeing what they're made of. What is their genetic code? All right, fast forward to 2002, Giovannoni et al. Had taken a small gene, 16S rRNA gene, we'll get a little bit more into that, and they wanted to say, let's go identify all these cultures. Let's use Sanger sequencing and let's do it. And they came up with a brilliant technique where they isolated these microbes. It's like single cell isolation. You just keep kind of like taking the colony you want, spreading it on a plate, taking the colony, spreading it on a plate. You get an exenic. It's like one microbe. You extract all the DNA fragments from there, and you run it through something called polymerase chain reaction. So this is just a very fancy way to say, like, control, copy, paste, like a million times. And so essentially what you end up getting are these target genes in the millions. It's exponential. It's unbelievably brilliant for a technology. And so what we're doing is we're taking this fragment and we are nesting, or we are like flanking the DNA sequence to target it. We are like finding very creative ways to target these sequences. And so they're roughly about 1,500 base pairs. And what this ends up yielding us using a DNA polymerase are the sequences that we can clone into plasmid vectors. I'm not going to get into the depths of this. I don't think it's part of the talk, but lo and behold, we end up with our bacterial strain. So now we can go, this plate is this strain of this bacteria. And it was groundbreaking. And it was 2002. Fast forward to 2006, Sogan et al. Leveraged PCR, and they basically said, let's look at all the microbes. Why are we just focusing on one or two? They didn't actually say that, I'm paraphrasing. But they went out, they took some seawater, they ran it through a 0.22 micron filter, which is basically the threshold at which most bacteria wouldn't go through. They extracted all the DNA from that community. They ran it through, but instead of going 1,500 base pairs, they cut it down to 500. They leveraged new technology. It was called 454 pyrosequencing. Again, not the purpose of this talk, but they developed this idea of microbial OTUs. These are operational taxonomic units. These are ways to classify individual microbes from a massive sample. And lo and behold, we have our microbiome. And thus, metabarcoding is born. This has taken the field by storm. We still use it today. We found much more unique and more precise ways to do this. But in short, primers, target, sequence, loads of amplicons, short read sequencing, and now we have all our taxa right there. So now you're all professionals. You can just go and get your PhD. What ends up happening from there is we develop these profiles, and we can look at like what all these communities are made of. Like, are they really rich in carbon metabolizers? Are they really rich in sulfur metabolizers? This is really critical information because we can leverage this, and I'm going to get into that in a moment. And so here's our eukaryotic diversity, this nice phylogenetic tree. And you got to ask yourself, is there one barcode that we can use to tackle all of these different taxa? The short answer is no. So what we do is instead we have different placements on this operon. So on the bottom there, it's a eukaryotic operon. I know some of this stuff's going to be a little deep for some of you and review for others. Not a big deal. Come find me later if you're confused. But essentially, we have our operon. Within that, we have our genes. And then we can use specific genes to target specific taxa. So our protists, we can explore using 18S or 6S. And then we have these spacer regions. Some of you that study corals, you might be familiar with IPS2 spacers. These are used for Symbiodiniaceae, the little dinoflagellates that give corals all that energy through the day, stony corals. And then we have IPS1, which is for fungi. And this is also used for prokaryotes, which some of you might have heard of 16S rRNA studies. And so we have these target barcodes, but they almost seem to be like very specific. And so we want to target our prokaryotic communities. We can use 16 sRNA genes. We can go for our bacteria. Certain parts of it, we can go for the archaea. If we want to target our micro-eukaryotic communities, our fungi and our algae, we would use the 18S marker. And then that leaves us with the viruses. And so viruses are complicated because they don't have a universal marker. And so rather we have to use something called metagenomics, metatranscriptomics. And I swear people just love throwing the name omics on things. But this way we can use a non-biased way. We're biasing, when we use PCR, we're biasing, we're targeting what we want. We amplify it to levels that we can basically track it. But metagenomics and metatranscriptomics are non-biased. And so it's really powerful. You can get so many different microbes from metagenomics. And then if we wanted to use metatranscriptomics, which is a fancy way of essentially sequencing the RNA. And we do this with using the poly A tails. It's really brilliant. I mean, there's a bunch of different ways to do RNA sequencing. We won't get into that. But what we're trying to ultimately get at is this active community. And the reason I use the word active is because in order for their RNA to be present, they have to be actively transcribing. They have to be like doing something. And that's even more powerful because so what if we get a bunch of microbes in our microbiome 16S study? We don't know that they're actually doing anything, or maybe they're just like broken up, degraded, and they're dead. So like at least this way, we know these things are doing something. And one of the more powerful ways to leverage this is our RNA viruses. And so now we've done it, we can sequence everything. So why not use metagenomics over metabarcoding? And so time and time again, I ask myself this, but it really just comes down to money. At the end of the day, everything comes down to money. You write grants, they're only going to fund so much. You have to be very, very conscientious about how you move forward as a scientist. And so metagenomics on host samples, so if I was to take a biopsy of a fish, a snail, a copepod, a brad, essentially what you're ending up with is a ton of that specific host. So it biases the sample. And so you have to do a ton of sequencing, very, very deep sequencing in order to get at all those microbes that aren't the host, right? This makes sense. And amplicon sequencing is cheap, and it just keeps getting cheaper every single year. And so metagenomics is typically done on environmental samples. I'm going out, I'm looking at my soils, I'm looking at my water communities. And so you're doing it without a host, a genuine like holobiome host. And so we can use transcriptomics on the entire holobiome, but if you look up there, little orange guy is our host, it's going to be pretty much the majority of it. And so if we look at the holobiome by composition, I'm going to use my little whole schematic here. Roughly, and obviously this isn't perfect, but roughly 90% of the sequencing reads come from our host. About 5% come from our dinoflagellate, the Symbiodiniceae, and then the remaining 5% is the entire remainder of the holobiome. All those other microbes, all that biodiversity. And so you can kind of, I hope the cogs are turning where you're like, that's pretty dumb. And so if you're trying to target that last 5%, you're wasting 95% of your sequencing, your effort, your money. And so metabarcoding is the way. So let's use metabarcoding to explore some microbiome structure. And my latest curiosity, which it's not my latest, this was back in 2002, but I wanted to look at coral microbiomes and whether or not they shift through day-night cycles, diel cycles. And so we have these natural circadian rhythms that kind of govern how our bodies perform certain tasks, how you generate insulin, how you metabolize glucose, et cetera, et cetera. So I wanted to look at the microbes. And so we're going on an adventure. There's Venezuela kind of at the bottom of the screen. And you have the ADC Islands. If any of you are familiar, in the middle there is Curacao. And at the leeward coast, which is kind of like the bottom part, well, in the middle, but on the left side, is a marine station called Karmabi. And so Karmabi isn't this oasis that's away from, you know, anthropogenic influences. Literally a 200 fin kick away is a resort. It's called Dreams. From there in the little blue box. But I wanted to find a site nearby because of the torture I was about to go through. So this is me sampling. So basically we found some collapsed structure on the seafloor. I don't know, maybe it was like an old dock or something, but it was like teeming with all these big coral colonies. And I was like, this is perfect. And so I talked to my advisor and we reluctantly agreed upon doing 3 replicate diel cycles. So in a lot of these omics techniques, triplicates is everything. And it's because we use a lot of central tendency. We need to get averages and get standard deviations or standard error. And so you can't really do that with 2 samples, bare minimum 3. And so that seems to be kind of like the agreed upon number. And so I started at midnight and I sampled at midnight, and then I went back again and I sampled 3 more samples at 6 a.m. and 12 and then 6 and then 12 and then 6 and then 12. And so I did this 3 times over, just me and my labbing. And so what we wanted to do, we ended up with 36 samples, and we wanted to sequence both 16S and 18S. And it's because we wanted to look at as much of the holobiont as we could with the funding I had as a PhD student. And so my lab mate looked at me and he says, "You want to dive every 6 hours for 3 days straight?" I'm so happy I had this guy because he was so down. Here's us. I think this is like 6 p.m., but obviously I couldn't get videos of the ones at midnight. But essentially, we were going out there and we were just getting triplicates and getting water samples. And I noticed the dive team's like, "You're looking pretty stupid right now." I promise you it was safe. I was very, very close with the dive office, and we made sure to dot every I and cross every T. And some of you might be thinking, "Man, you probably really screwed that coral up." So day 1, day 3, a lot of samples taken from it. I understand that. I went back 6 months later and the coral had almost completely regenerated. I mean, it wasn't perfect, but I mean, probably in another 6 months, it was like probably good to go and ready for another spawning cycle. And so recall metabarcoding. We're trying to look at this like microbial community exploration. We're trying to look for core microbial members and these like funky critters that are constantly there all the time. And in the context of my stuff, at every 6 hours, who's showing up at dusk, who's showing up at midnight, and why. And so you can do this. Don't worry about the plots. The information in there is not too important, but it's just to give you some context about what we can do. And so we can look at alpha diversity, which is like Shannon, Pylus, Pylus evenness, Simpson, inverse Simpson. And these are ways to look at the community as a whole. How diverse is it? How dominant are certain taxa over other taxa? We can look at beta diversity, which is like how close are 2 samples to one another in terms of their composition. We can use things like Bray-Curtis, Euclidean distance, Manhattan distance, Aitchison distance. I'm not going to worry. And then we can also look at the community composition. Text is really small. Don't worry. You don't need to look at that. But what these are showing, and I'm going to have an example right after this, is specific genera of microbes and what they're doing time over time. And I can use this because I have replication and I can show these microbes that are popping up maybe at specific time points. And we can run things like network analyses. We can do differential abundance. We can say, using statistics, this specific taxa is showing up at this specific time consistently. It's probably pretty important. And so zooming in on that one that I said, don't worry about looking at. So this is our eukaryotic genera, and I'm not going to bore you with all the different types, but essentially it looks pretty stable, right? There's not a lot of change happening there. So at the top there, it's pretty much like your diatoms and your algae and some ciliates, phyllacers, some ciliates there. And then on the bottom there, where it says Swetziales, Symbiodiniaceae, Durstinium, Braviolum, those are the algae symbionts that give the host like 90% of its photosynthesis, its carbon energy. And then in the middle there, we have like a proportion. And so like the proportion of our Symbiodiniaceae to every other microeukarya is like 50-50. It's pretty wild and maybe a bit of a waste of sequencing. And so when we look at these network analyses, what we're trying to do is get an approximation to who's co-occurring with who. It kind of gives like a proxy to microbial interactions. And so we've got our Braviolum right there, right? That's our symbiont. And then I highlight a couple taxa that you can't really see. It's not really important for this talk. I mean, so I'm just showing you how to find them. But ultimately, what we are seeing are taxa that are involved in biogeochemical cycling, immune responses, and ultimately like host protection. And so some of the major findings that I found really exciting were typically at dusk. And I'll explain why this made sense unless you get there before me. We found some glycogen scavengers. These are like little microbes that look to like grab glycogen and just hold it for some reason. And then we found a lot of poly and monosaccharide metabolizers. And so what you're seeing here is actually microbially mediated carbon cycle, which is pretty cool just from some 16S data. And so these are the types of things that we're looking for so we can make approximations towards to understand these systems a little bit better. But there are limits. There are limits to all of these different types of omics. We're still kind of in the beginning stages of metabarcoding, in my opinion. And specifically for this, we can typically only get down to genus, which is functionally important because when you get down to species and strain, this is where you start seeing things like really pick up on who's doing what. I like to use COVID as an example, even though it's a virus, but sometimes you get sick and sometimes you don't. Some microbes you get sick and sometimes you don't. It's the same flu that's going around, but some people are getting it, some people aren't. That is a strain level difference. So that is at the level at which we need to be exploring these communities to actually understand the functional potential of the microbiome. And so up here, I've got the small subunit of the 16S RNA gene, and Illumina, Illumina right there, it can sequence about 550 base pairs with paired end reading. And so 550 base pairs isn't really that much. And so if we can push that to something that has just been introduced, I mean, we've had long read sequencing for a while now, but it's called high throughput, fancy. But what we can do is we can get from 500 to 1500 to 4500. We can sequence entire operons. And this is really, really powerful because now we're getting that strain level resolution. And that will help us to get to these critical understandings of what these like rare ubiquitous microbes might be doing in the holobiont. And omics offers us to be able to reach really obscure environments. Think of the deep sea, think of space, the International Space Station. There was like an article about how there's microbes up there, but also like cryptic microbial diseases. And so understanding the variations between how people are getting diseased and biotech advancements. And so you consider hydrothermal vents. I think the deepest one that I could find on the internet was like 24,000 something feet. No one's going down there, but ROVs can. And so we can go down there, we can take samples, and we can preserve them and bring them back and sequence them and get a better idea of the biodiversity that's actually taking up those environments. This is a photo of like a disease lesion line on a coral. I think that one's, I don't know, it's really hard to tell the difference between coral diseases, but you can kind of see that white stark line and then there's like black stuff above it. And that is like really toxic sulfur and like these crazy secondary metabolites that are making it almost unbearable for certain taxa. But then you have these copiotrophs and these saprophytes that are just thriving. And lastly, I like to use Streptococcus as an example. I don't know if you're familiar with CRISPR-Cas9. You've at least probably heard the term CRISPR-Cas9, gene editing tool. And so that was found in a bacterium. It was this bacteriophage and it was putting in these repeats. And somebody was like, holy moly, we can totally use this and snip up DNA and maybe change someone's life. You know, maybe we can find a way to alter a part of the genome that would be coding for a certain hereditary disease. And these are evolving. It's not just CRISPR-Cas9, there's Cas12, there's Cas13. And so we're constantly advancing our ability to understand this stuff as we dive deeper into these different microbes. So you're thinking, sweet, we just looked at the microbiome, but like, it doesn't really give us that much, right? Our coral, we saw some taxa that were showing some diel rhythmicity, some periodicity. And so what we try and do in omics is actually layer different types. And so what we can do is layer that with transcriptomics. And so now we have the microbiome, we have an idea of what some members are doing, but we don't know what the host is doing. So we can sequence all of the RNA from the host, because remember how I told you it's like 95 or 90% of the total reads, and we can look at what the host is doing through time. And this is visualized through gene expression. And so this kind of came about because you can preserve samples for both DNA and RNA at the same time. I didn't have to go do this stupid thing over again. It was all the same samples, same preservation tools. And so I collected 36 samples of this coral and I sequenced it for the RNA to look at, immune pathways. I want to look at how it's dealing with biogeochemical cycling, how it's using repair and rest mechanisms and recovery, and what it's doing through the onslaught of UV that it faces through the day and at night when it goes hypoxic because all the oxygen is used out. And so what we can use is this tool to get real-time gene expression. We can see this through another PCA type plot, which is showing how samples are related to one another. It's called gene dispersion. We can look at gene ontology, so these transcripts that we're annotating, we can annotate them to known databases and genomes and get an approximate of using orthologous genes. We can look at PEG protein pathways to get an understanding of what parts of these pathways are being activated. And one of the unique aspects of my study, I wanted to look if I could find specific gene transcripts that were showing consistent peak to trough day over day at the exact same time, because that's the coral turning the light switch off, turning it back on again, and turning it off again. And that is indicative of circadian entrainment. And so much to my surprise, when I did this is a PCA plot, essentially it's just some space where you calculate relatedness between samples, and the samples literally plotted like a clock. I was kind of blown away by that. And so one of the unique aspects of doing this type of RNA sequencing is, remember how I said 5% roughly goes to the symbiont? Well, I can also look at the symbiont's gene expression too by deep diving into their sequencing reads. And I didn't see a lot. It didn't seem like time was really having that massive of an impact. So it didn't seem like it was overly regulated, but its transcriptional control wasn't regulated through circadian entrainment. But this is called a heat map. It's basically scaled gene expression. On the y-axis there, we have all gene transcripts, like thousands of them. We're just looking for color differences here. It's not that fancy. And then on the x-axis is our samples, and they're color-coded by time of day. So think like the blue ones are midnight, gray is dawn. But what we're seeing is a lot of the natural clustering is actually happening at night and into the morning. And so I started thinking like, whoa, maybe there's like something happening with early light cues, and it's maybe like waking up a little bit. Maybe there's certain light wavelengths that are triggering some cascades to upregulate a bunch of things. And so we can call this cellular activation. And I can do this too with the symbiont. And so when I was looking at the symbiont data, it didn't show this like really aggressive light and dark situation, but it did show clusters of genes that were very specific. That's clustered by itself. And so it clustered based on time point. I thought that was pretty wild. But I wanted to go one step further. What are these transcripts doing? They're just little lines that are colored blue and red. Who cares? We can use gene ontology. Don't worry about the lettering. I overlaid it for you guys. So essentially what we're seeing is based on time of day, we're seeing the coral do different things. And I think that's really important because no one's really done this and gone out in situ and actually recorded what these animals are doing. And so what we are seeing is during the night, it was going through this like rest and recovery stage where it was going through cellular repair. A lot of the organelles, the proteins that are being damaged, misfolded through that onslaught of UV were being broken down. And then towards 6 a.m., we're getting RNA synthesis, biosynthesis. So it's rebuilding things and then it's activating it. So it's basically like priming a cellular toolkit so it can take on the day. And then as we go towards noon, which is yellow, we get phosphorylation, dephosphorylation. It's basically the corals dealing with all of the photosynthates that the dinoflagellate is throwing at it. So much carbon, so much sugar. And then towards the evening, there's a lot of like metabolism, regulation, fine-tuning controls of all of these proteins that are helping the coral manage all of this oxygen, all of this carbon. When I was looking at the symbiont, there were a lot less genes. There was pretty poor annotation overall, but again, it was 5% of the overall sequencing. The fact that I got anything, I was pretty stoked. But midnight, we were seeing cellular division, so these things are actively replicating. 6 a.m., we're seeing photoprotection. It was starting to like prime itself to prepare for this onslaught of sunlight. And if you're familiar with coral bleaching, have you guys heard that term before? Yeah. So what happens is the photosystem 2 breaks down, and that happens in the thylakoid membrane because of these things called reactive oxygen species. I'm not going to get too deep into that, but it's literally protecting itself against that. I mean, at least there was traces. You can only go so far with gene ontology. You know, annotations are pretty complicated. But, and then when we get towards midday, we're seeing metabolite transport. And so this was indicative of it overproducing sugars and needing to shunt them away from it. So it's shooting it out into the cell, and they're being transported all through the coral. I just thought this is such a wild picture. And then towards evening, we're getting a lot of like metabolism and recycling of these components. When we're looking at the protein pathways, this one's pretty stark. So basically on the left there, these are significant, they're called alluvial plots. They're mainly just, it's like a narrative Sankey plot. It's a way to like just show color and size. So you don't really need to know too much, but like a lot of gray means a lot of things are happening and it's super significant. So at 6 a.m., we're seeing tons of genetic information processing, translation, transcription, chromosome, protein folding, sorting, and degradation. Pretty much all indicative from the gene ontology I was just telling you about. And so as we do more of these different types of analyses, they give support to the ones before it, because you can't really just trust one analysis. You got to do a ton as scientists. That's just kind of what we do. And then for breviolum, I didn't see a lot of time-dependent significance. It didn't seem like it really cared at what time it did any of these tasks. It just kind of exists. And so that final bread and butter of this experiment was trying to isolate specific transcripts that showed these peak and trough in amplitude and these functions that it's undergoing. So without reading too much into this, essentially what we found it, about 50% of the transcripts recovered and annotated in the coral were rhythmic. These animals are super tightly controlled transcriptionally through light and dark cycles. And so this is all about to be submitted hopefully in the next week. That would be pretty cool. But the main takeaways of this is the first time someone's done this. And I'm really excited to share it because it's hopefully going to influence someone to build upon this and do a better job than I did. But in summation, we see this coral is highly regulated in its transcriptional control. The symbiont also had diel rhythmicity, this like periodicity, but it was like muted. It was a little more subtle than the coral. And then the microbiome was pretty stable overall, but when you really dig in with those statistics, you can pull out very specific taxa that have really important functions. And so this kind of hopefully convinced you that we need to integrate multiple types of omics on top of one another in order to get the whole picture. We can use one type and get a snapshot, but the more you can do, I just showed you, like you start to unpiece this puzzle, and it's pretty amazing. And so using the host transcriptomics and gene expression and seeing how it was influenced by diel cycles, the microbial community and those functional annotations from the metatranscriptome and the metabarcoding results kind of starts tying everything into this holobiont function, its structure, its function. How is it dealing with I mean, we're still deciding whether or not corals have immune systems, but the immunity, programmed cell death, repair mechanisms, biogeochemical cycling, and these are really critical because we do this on a day-to-day cycle. People have studied humans to no end in terms of how we are deeply entrained by circadian rhythms. And we can leverage this information as a baseline. And if we know what a coral is supposed to be doing, or a human or a snail, then when we see an outbreak of a disease, we know it's weird because we have something to compare it against, right? We have the direct comparison of what is abnormal about this thing. I think that's really important because a lot of baseline studies don't get funding. And it's really stressful because you're supposed to just like figure out this puzzle, but all your pieces are underneath the couch and in your vacuum. And it's just really frustrating. And so by taking these big systems, these holobionts, the structures, the functions, it's like this system level biology, and we can make these comparisons and make really informed hypotheses and understanding of these systems. And so today I just talked a little bit about genomics and transcriptomics, but there are so many different types. And like I was saying earlier, people just throw omics on the end of anything these days, but it's really understanding chromosomal level variations and understanding methylation and proteomics in terms of like who's there, what analytes and hormones are present, metabolomics. And so integrating all of this together, we can really push the field forward in understanding baseline and comparing it to make like an actual understanding of what disease and abnormalities look like. And so I hope I convinced you how to find microscopic beasts. Thank you.
Thank you so much, Brad. We're now open for questions.
Hi, thank you very much. That was awesome.
I am thinking about, so we have like symbionts, coral hosts, and the holobiome or the microbiome. My broad, probably unanswerable question is, who's at the steering wheel? And does it change from day to day? And my more specific question is, did you compare like within an individual, are they influencing each other? Like maybe like upregulation in the symbiont is affecting the host. I don't know if you did that analysis or you have an answer.
I love the first that question. The reason is we anthropomorphize, did I say that word right? We look at the host so much and we never focus on everything around it. We are so host-centric in everything we do. And I love the way you word that, because who is at the steering wheel? We don't know. And so people have been looking at humans, gut microbiomes, and there's some fascinating things that have come out of that. If your diet is composed of like fats and sugars, you grow microbes, your gut flora, to reflect that environment, and then you crave it. So those chocolate cravings you're getting, those cravings for fried chicken or whatever it is, that's being driven by microbes. That's crazy. I don't actually know the answer, to be honest. I don't know if anybody does. But I think it's a really important thing to consider, because we are so host-centric and we think that we're in control, but I mean, we're mostly microbes. It's kind of trippy. What was the second part was whether or not symbionts are impacting the host, like its transcription is.
Yeah.
I think they work in a symbiosis. I mean, we can go all day about symbioses and parasitism and all that stuff. We don't know who's in control there either, whether the symbionts are being jail celled or whether they are kind of leeching or parasitizing off the host. But you kind of see they are working together. So whether or not it's kind of like a Stockholm syndrome where they're just like stoked to be there and just kind of doing stuff anyways, I don't know. But at the end of the day, I think it's just important to understand the system-wide co-evolution, because over time they've grown to live with these things. And if you have something that can do a function for you through millennia, millions of years, you're losing that in your genetic code because it's redundant. You don't need it anymore. And so you actually lose. And so people study this really, really deeply in microbes and like protists, because you end up losing cellular or genomic information on how to do specific tasks because you have an association with something that can just do it for you. I mean, it's pretty smart.
Thank you very much for your talk. So my question is more based on, this study was done in an area where there's like pretty equal daylight year round. Would that change in areas where there's more like seasonality? Would there be less of like a entrained relationship between like the field cycle?
I think I know where you're going with this. So you're asking like Finland, for example, where there's like 24-hour darkness or something like that, right? We don't fully comprehend how like you gotta think these organisms are sessile. They're not moving. They're not getting up and moving away from Finland, right? So the concept there, I think, changes between your life survival and history traits. As humans, we have done tons of research on whether or not we are impacted, and what they'll just throw somebody in a dark room and kind of like see what happens to them. They do this with mice, they do this with all sorts of organisms. No one's really done this. Like, it has been done in corals. I shouldn't say that. It's lab-based. It's really like all the variations controlled. I think that's why I like this one. It was in situ. It was like literally in the field. There was a spawning event that kind of happened, a reef-wide spawning event that happened at the end between pearl stars and sponges. And so it's just chaos, right? But if I can get patterns like that in a chaos, that's pretty cool. And so I don't know if you need lab-based equipment to do this kind of stuff. So to answer your question, I don't know how corals are going to respond specifically in the field, but in the lab, we do show that like they still function through darkness until it goes away. So it's like a lagged effect. And I think that happens in humans as well, where like we still produce the components we need to at specific hours of the day until a certain point of light or dark being removed. And then over time, we lose that. Does that answer your question?
Yeah, cool. Thank you.
Hi, thanks. I was curious about the symbiodinium not having that sort of pattern to its functions when you expected it would. And I wonder if you could talk about how much, like what you can discover depends on having enough things for that to be mapped to, and whether, yeah, I'm just curious about sort of how that affects the entire balance, I guess.
So basically, if I'm understanding correctly, it's this, like we didn't see amazing patterns in the symbiont, and whether that's like a bias in the technology or a bias in the organism, is that right? So 5% roughly of my reads went to it. If I was able to isolate or have some specific way, like I could probably find a way to sequence deeper and get more information. The more information I have, as long as I'm doing genome guided annotations, theoretically it should get better. I'm working a lot with protein language models right now. So we're finding machine learning ways to predictively assume what the proteins are. Some of the preliminary spoilers are it's like 10 to the power of 6 more significant. And so it's really, really powerful, but I just didn't have time for this paper because I got to move on. But 100%. And I think if I could have sequenced deeper, we would have seen really amazing patterns. And I urge you to look up Frank Alward, I think Mohamed Monaruzman. They did a bunch of dinoflagellate work, planktonic dinoflagellate work, where they saw incredible diel patterns in them. And so in the discussion of the paper, I do go into that a little bit.
Also, can you talk to like half of RIB has things they probably want to ask you. We have 2 students who are trying to figure out an unknown, either parasite or commensal, one metazoan and one protist, and somebody doing diel patterns in zooplankton. So just a request to set aside time. Fascinating overlap.
Yeah, let's chat after. Absolutely.
And we should mention that Brad will be here until Friday. He will be spending some time with the shark biology and conservation class, I believe, on Thursday.
I also plan on going to the tide pool thing tomorrow because I want to look at tide pool critters.
Do we have other questions at this time?
We got one over here.
Oh, thank you.
Was there any online?
We're checking.
Okay.
Hi, thank you for your talk. I was just wondering, when sampling, when doing this analysis, did you have to account for the effects of the coral stress from the sampling, like in terms of the holobiont or in terms of the transcription?
Absolutely. But it would have been reflected on a day-to-day bias. And so I controlled for day through a lot of my analyses and saw no patterning, except for some, but it wasn't enough that was impacting the day-to-day functions that we were seeing. And so if you look at the functions and separate day by day, because it's still triplicates through each day, we still see the same things. And so at midnight, I did see a lot of immune-related, stress-related, but they always boiled back to hypoxia. And so I'm going to dive into that really, really quick. Corals, they have the symbionts that they're associated with during the day. They're overproducing through photosynthesis, and they're at like 250% oversaturation of oxygen. And then as you go through the night, the holobiont is using up all of the reserves of that oxygen. And so you do end up at a point where you're hypoxic, or at least nearing hypoxia. And so the corals and everything in there is starving for oxygen. So we start seeing more hypoxic tolerant taxa start showing up. But essentially what's happening is proteins are folding and misfolding, and organelles are being destroyed because they can't handle the hypoxia. So that was the only stress that I was seeing through that. But that is a really good point, because you saw it was like pretty destructive. I mean, at the time, so if I was a coral, I would have been pretty upset.
There's no line.
There's not much. Jeff Guess is asking, does your analysis suggest when your corals are most likely to feed?
In terms of, I'm assuming they mean heterotrophically. That's a good question. I didn't see anything other than like, so there are specific Go terms, gene ontology terms that are like response to stimulus. And typically what this relates to is like a tentacle moving. So there's some like cue that's responding. So maybe like something bumps into a tentacle and it's like, it grabs it and it eats it. So maybe really hard to make that one-to-one, like this is happening because it has a response to stimulus, because the stimulus could be anything. Right? It could be hypoxia. It could be responding to low oxygen. But I had talked to some colleagues who said they've seen a similar thing when they're feeding, and they take samples when they're feeding, and it does end up being this like response to stimulus, but I don't know, to be honest. It wasn't like heterotrophic feeding, so it's a really good question.
Other questions in the room? Oh, thank you for the presentation. And I'm just wondering for the design of the experiment, right? And I think one major thing you mentioned is the limitations of resources. So I was wondering for the design, what makes you collect sample from this particular coral colonies, and why did you do it this time? I just want to know what's the overall thinking behind that design of the experiment process.
You're asking a guy that wasn't really thinking straight. Okay, to be honest, my advisor asked me to do this in Florida, and it never happened, COVID, and it was just confusing. And then all the colonies were dying. So we finally went to Curacao, and it was like this perfect opportunity. So we went on some like preliminary dives, just like scout missions. And we were outside of this like resort, and like I looked down, it's just like, it was perfect. And I was like, I could go to a coral reef patch where there's a ton of different corals and choose one at random, or I could find this like really obvious giant concrete structure, because after day 2 you're pretty loopy. By day 3 you hate yourself, and like you can't see at night. When there was a spawning event, the entirety of the ocean was like milky white, so you had no visibility. And so I went with the easiest way to not screw up, I guess. And so the structure, like you're swimming, you kind of have a general idea, 200 fin kicks, and you should be there. You just kind of shine the flashlight and you look for this giant structure. That was the rationale. The specific corals, it was kind of haphazard. I just wanted to look for the biggest colonies to do the least damage possible, because if it was a smaller colony, I probably would have just destroyed the whole thing. But there aren't massive, massive boulder corals in that area. And so it was just kind of like the resources I had and the time frame that I had, and like everything had to work out perfect, right? Like if I had an earache or my partner started feeling sick, like the whole experiment would have died. And so like everything had to be perfect. So I went with like the path of least resistance, essentially. The 6 hours later, after I finished it, a swarm of sea wasps came in, so that would have destroyed the experiment. So like literally a shift of 6 hours and my whole experiment would have been toast. So it's kind of, yeah, it aligns with like just haphazard sampling, I guess.
Yeah, great talk. You raised a really interesting point though. So you were doing this work off of a hotel. And as we're learning, right, the rules of urban marine ecology can sometimes be quite different than in a sort of, I mean, I don't want to say pristine, that doesn't exist anymore, but in a ecosystem that has lower levels of impacts. Do you think you were
seeing any signals of that in, say, the expression of the microbiome or the transcriptomics?
Is that in relation to the diel cycles specifically, or just in general? Okay, because one question I thought I was gonna get is artificial light at night. That's something that people think of quite often in marine realms, which maybe, I hope I didn't just open a slew of more questions, but I don't know how much it is one coral sampled several times, but hopefully through enough replicated sampling. I didn't mention, I did this with 3 more different species. So there will be a much bigger paper in roughly, hopefully a year, but gotta publish right away kind of thing. And so this one species in particular, I didn't see any like obvious effects that it was sick or any obvious effect, because you have to do some pretty deep literature review to understand what you're even looking at. And so nothing that I saw was indicative that this coral might be sick or visually impaired by any stretch. But you raise a good point in that it's right off a hotel. And so we know when people are swimming, I mean, it was still probably about 12 to 15 feet deep. And so I don't imagine a lot of kids are getting down there and kicking up their fins and stuff like that, but that is a real thing to think about with patch reefs off of resorts, because I've ever been to a resort, people are literally standing on corals. And so absolutely that could have impacted it. But I chose one that was kind of deep enough that you kind of hope people aren't finding a way to get down there and punching it or something. So I'm assuming it was probably pretty healthy. There's always happy hour.
Well, and so one of my questions, and it kind of relates to what Jared was asking, is with all of the and maybe I'm wrong on this, but my understanding is that there's sunscreens that are now impacting corals, and whether there's already stressors, particularly in more urban type habitats, that might be impacting some of what you're seeing, also because of the impact that it would have on diel cycles because of light.
It's a bit of a misconception, the whole sunscreen thing. That work, I don't want to talk badly about it, but this concept of oxybenzone and its effects as a slick, preventing light penetration, reducing the photosynthetic efficiency, I don't know if there's a lot there. Am I saying lather yourself with sunscreen and just go dive on some corals? No, obviously use your head, but I don't think that is, we're facing pollution, we're facing sedimentation from dredging events, we're facing year over year more frequent bleaching events, we're facing new epizootics that are popping up because of poor sewage. These things are facing so many problems. I don't think sunscreen is much to worry about right now. I think we need to focus on our CO2 emissions. We need to be more conscientious about our daily uses of just waste culture. I think there's just a lot bigger problems to deal with. Does that answer the question?
Yeah, thank you. So, well, no, our time is up at this point, but as I said, Brad's going to be here for most of the rest of the week. I want to thank you so much for such thought-provoking presentation. I think it was really interesting also to talk about the realities of field work, especially with these particular groups of students that are here at the island right now. And so I'd like everyone to please give another round of applause for Brad. Just as a reminder, we have 3 more rock talks in this year's sequence. Next week is our very own Taylor Lindsay, who will be speaking on some of her PhD research. Tune in Tuesday, 8 o'clock, Rock Talk. Thank you for your attention this evening.
Thanks, everybody.